Hello, family. How are you? We're back again. We're doing biology. I hope you're good. I hope you're good. I hope you're good. Today I came back. Now I just came back with so much energy, right? Anyway, so we're doing the second topic of combined science, the biology section. We're looking at cells. The chloroplast today, right? These ones. They will also be mitochondria, but at your level right now in combined science, you don't talk about the mitochondria that you find inside the cell. So both cells have mitochondria, but at this level about them. But you are only going to see the mitochondria when you are given questions with this type of cell. So the mitochondria or some other type of cell like the sperm cell. So this cell is called the root hair cell. So it is found at the ends of the plant roots. Its function is to absorb water and mineral salts or minerals from the soil, right? How is it adapted? What are the features that enables it to do the function, right? Number one, these cells, have a large surface area for water intake. Imagine if they were just rounded, right? And they were small and compact and everything, and this thing was coiled together and just rounded. It means there will be less surface area for water to be absorbed. But this one, because it is extended like this, means water can come through here, can come through all of this space, right? So it means there's a large surface area for water intake, which is its function, right, to absorb water. They have and minerals from the soil. Number two, they have a large number of mitochondria for respiration to become more active. So mitochondria, this is the site where respiration is taking place inside the cell. So it has many mitochondria so that it can have more energy to become more active. So that it can perform the function of absorbing the minerals from the soil. Number three, they have a concentrated vacuole to support absorption of water by osmosis. So the vacuole is very concentrated. There's a lot of sugars, right? So water will naturally move by osmosis from a place where there is a high concentration of water molecule to a place where there is low concentration of water molecule. And in the cell, there will be a low concentration of water because this one has many sugars, right? So water will move from outside the cell into the cell. So that's basically what we're talking about here. Okay, so you just pause the video and go ahead and just read all of these things. So you already know that this cell is being found on the roots, right? So it doesn't need chloroplast to trap sunlight for photosynthesis because photosynthesis is not happening because this is usually under the soil, right? So there's no photosynthesis happening under the soil, right? So and there's no light getting there, so there's no need for chloroplasts. I don't know if you got me. Please also just write down if you have any questions and just send them or put them down in the comments and then we'll just address them. Then we have what are called palisade mesophyll cells, right? So just to give you a brief overview, when you have a leaf, right, and you cut it through, right, this is what the inside of a leaf looks like. This is the top of the leaf. This is the bottom of the leaf, right? The top we have a cuticle and we have a layer of cells called the upper epidermis, and these are the cells that we want to talk about. They're called the palisade mesophyll cells, and these ones are called the spongy mesophills. This is the lower epidermis, and this is the lower cuticle. Then here we have what is called the vascular bundle, which consists of the xylem and the phloem. These are basically pipes or tubes. This one is basically a tube, right, that carries water and mineral ions, and carries the food in the plant, the phloem. So this is the palisade mesophyll, right, cells. So let's look at one of them, which is this one. It basically has the same things for the plant cell, everything, but it's just that it has many chloroplasts. And why? Because it's found on the leaves, right? And it needs a lot of chloroplasts to be able to absorb the sunlight for photosynthesis to happen. So it's found beneath the epidermis of a leaf, which is this one, it's the upper epidermis, right? And it consists of one or two layers of closely packed, long and cylindrical cells containing chloroplasts, right? A cylinder is something like a can of beans, right? That's the shape of a cylinder. That's what we're calling. So we're calling the cells as cylindrical. Their function is to facilitate photosynthesis. So to make photosynthesis happen, basically, they contain numerous chloroplasts to allow maximum absorption of what? Of light. So that because so that photosynthesis is happening in the leaves mainly, right? So it needs to have a lot of chloroplasts. That's why leaves are very green, because the chloroplasts are green. So the whole cell will look green. Okay. So let's look at the another cell, the sperm cell. Right? This is what the sperm cell looks like. This first part here is called the tail. This middle part is called the middle part, or the middle piece, and this is the head. The tail helps the sperm to swim so that it can go and fertilize the egg, right? Then this middle part has a lot of mitochondria, the ones we've talked about back then when we said, when we talked about the root cell, right? This is where respiration is taking place, and we need a lot of mitochondria because respiration releases energy, right? So this sperm cell needs a lot of energy to swim so that it can be fertilized, right? The head here has a digestive enzymes which breaks down the outer membrane of the ovum, right? So the ovum is the female sex cell, or the egg, right? So this acrosome will contain enzymes that breaks down so the sperm can penetrate the egg and fertilization can happen, which is when the nucleus of the sperm fuses with the nucleus of the egg, or of the ovum, right? Then the middle piece has a lot of mitochondria to provide energy for the sperm to swim. So this is not in the syllabus, but you need to know it because you're going to be given questions that might be related to that. If you want to pause here and take a bit of some notes, please feel free to do that. Feel free to pause and take a bit of some notes right there. So let's talk about magnification, right? We want to be able to calculate magnification and size of a biological specimen using millimeters as units. So for example, when you see a cell, remember, you can have a drawing of a cell, right? But that cell has been magnified because you can't see it with your own eyes, right? It has to be magnified. It has to be zoomed out so that it becomes a huge image that you can actually see. So if we magnify something, let's say times 30, right? It means the size of that thing has been multiplied times 30. So this size, the thing is appearing 30 times bigger. So for example, if you want to know the size of the image, right? You say the actual size. If you know the actual size of the image times the magnification, that's how you get the image size. So for example here, let's say your actual size is 1 millimeter and you multiply it by 300 in terms of size, right? It becomes 300 millimeters. So the image that you draw will be 300 millimeters, but the actual was 101 millimeter, but we have zoomed it out times 300, which becomes 300 millimeters, which is the image. I don't know if it's making sense. So you can use this. So if you're given actual size and magnification, and then you're told to find image size, it just says actual size times magnification, actual times magnification. If you're given actual size and you're told if you're given actual size and image size and you're told to find how many times this thing has been magnified, you say magnification is equals to image size divided by actual size, right? So if you're trying to find this, you say this one times this one. If you're trying to find this, you say this one times this one. If you're trying to find this, you say this one times this one. This is making sense. So for example here, we have been given an image that is 30 millimeters long, and that image has been magnified times 30 times 3000. So if you're asked to find the actual size, you say image size over magnification. So actual size is image over magnification. Image is 30 millimeters, magnification is 3000. So if you divide it, you get 0.01 millimeters. So that will be what? Your actual size. But it has been multiplied 3000 times, that's why it's appearing as 30 millimeters. I hope you got this one. If you have any questions on this one, please let me know. I'm happy to help. And The length of a cell drawing is 20 millimeters. Calculate the magnification if the actual length is 0.02 millimeters. So you've been given the length of a drawing, which is the image, it's 20, right? And you've been told that its actual length is 0.02. You have this one, and you're asked to calculate the magnification. So you're basically just saying image over actual, which is 20 millimeters divided by 0.02 millimeters. Then the other thing to note that is very important is when you're calculating these things, right? So I want to see your answer. Please get me this answer and send me so that I can see your answer and see if it's correct. So when you're calculating these things, make sure everything is in millimeters before you proceed. Make sure everything has been put in terms of millimeters, and then you can freely proceed. So that's it on the topic of cells. So we'll be moving on to movement into and out of the cell in our next topic. Please watch this video over and over and over and over again, and you'll get it. So that's me. I'm out. Have a good day. Ciao.